Good evening, everybody. It's a pleasure to be here, and it's nice to see so many familiar faces. Go ahead and go to the next slide. And the next slide. So I have a lot of colleagues here this evening. I'll just start with myself. So I'm a medical oncologist. I take care of patients with breast cancer. My specialty is in all types of breast cancer, but I have a special focus in patients with metastatic breast cancer and early phase clinical trials looking at new cancer drugs in patients with breast cancer, including triple negative breast cancer. I am the Joyce M. Brown Chair for Developmental Therapeutics and Women's Cancer, and I'd like to give a special thank you to the Brown family for making that possible for me. And with that chair, it really allows me protected time to make sure I have the effort to dedicate to my research work. I also co-direct our phase 1 program, which we call our POEMS program now. And then we'll go ahead and go to the next slide, and then we'll let Dr. Borges talk too. So I just wanted to start with a special patient that really provides motivation to me on a daily basis, and this is why we all do this work. My patient Sandy was diagnosed with triple negative breast cancer. Triple negative breast cancer is an aggressive breast cancer subtype, and she was treated with standard therapies, but unfortunately suffered a recurrence of her cancer, which was not responding to chemotherapy, which was really the best option that we had. And so I met her, and she was interested in a new approach and was interested in early phase clinical trials and new drugs that would be investigational at the time, but could be potentially impactful and effective for her cancer. And so she enrolled on a phase 1 clinical trial, which is the first time this therapy was used in people with cancer. And she had a complete response to therapy and is now in a complete remission almost 5 years later. And so I see her now every 6 months, and she has no evidence of cancer, which is truly remarkable. Unfortunately, not everyone has the same response, but it's these exceptional responders that really help us to try to develop these new therapies and try to broaden the impact so that more patients can have these kind of responses. And so my focus is really taking new treatments from bench to bedside and really ensuring that there's early phase clinical trial options for all patients with metastatic breast cancer. We'll go ahead and go on to the next slide. So I wanted to just talk a little bit about a project that we're working on now that's heavily funded by philanthropy. And so we know that immunotherapy can be used to help your own body fight cancer, and it's proven and effective in many tumor types, including melanoma and lung cancer, and also in triple negative breast cancer. But unfortunately, not everyone will have the same response that Sandy had. And so we've used preclinical models in combination with Julie Lang, and we have a humanized mouse model. So it's basically a mouse that has a human immune system and is not immunocompromised. And so we've been able to investigate how immunotherapy works there and use that to support a study looking at using radiation as a way to potentiate this activity. And so this is a study that Christine Fisher is partnering with me on. She's one of our radiation oncologists, and this study is funded by donor philanthropy from the Cancer League of Colorado and also from independent donors to our Women's Cancer Developmental Therapeutics program. And now we're applying for NIH funding to support the correlative work. And so this is just an example like I think that the chancellor or our cancer center director was talking about, we can use the philanthropy support to start an idea to really get it going. And then we can leverage that funding to get external funding through NIH or DOD, really to make all of these things possible. So we thank you. We'll go ahead and go to the next slide, and I'll give an introduction to Dr. Borges. So you saw her picture on the earlier slide. So Dr. Borges is a professor, and she's our deputy division head and heads up our clinical breast cancer research program. And so Ginger, I'll let you take it away.
Someone was going to have to start talking while muted tonight. I filled that role. So thanks, Jen. I appreciate the introduction. I have been at the University of Colorado for 18 years, and I began my research career a little over 20 years ago. And my research focus in life is young women's breast cancer, and I run a couple of things at the university, one of which is the Young Women's Breast Cancer programs, both clinically and translational research. And so when we do our clinical trials and select for these clinical trials, we often will have a focus towards problems that are very specific or dominant in young women's breast cancer, though they happen in all women who face breast cancer as well. And so tonight, one of the things I thought I would focus on across the research we've done over the years is kind of a fun story because it's a Colorado story. So I thought you'd enjoy that. So many years ago, over 10 now, came across a compound that at the time was known as Array 380. And it was showing very interesting promise in the bench. And Array is a biotech company up north of here that we were partnering with. And from there, was able to lead 2 of the national phase 1B trials that got this drug moving forward in time. It ended up being sold to a couple of companies, so you'll see some name changes. Eventually, it became what we now call tucatinib, and it entered into a randomized phase 3 clinical trial where it proved itself to be stunningly successful. It has become a practice-changing medication, the results of which were released only about 18 months ago. So big news in our world, and it was great to be part of it from ground zero, entering human beings until moving forward. But as we got comfortable and familiar with this drug, and I really began to appreciate its ability, we were able, through philanthropy and through help of donor funds in the Cancer Center, to be able to explore what we wanted to do next with tucatinib as an important drug. So one of the issues in young women's breast cancer is they tend to get an increased amount of HER2 positive breast cancer and also an increased amount of breast cancers that are estrogen receptor positive and HER2 positive. And it's a particular conundrum because when you have both markers, neither the estrogen blocking drugs nor the HER2 blocking drugs work quite as well. And also, these are tumors that unfortunately like to form brain metastasis, and that's a very big problem. But with the advances I'm going to show you here, we've been able to make some very serious headway. So we were able to launch our own study based here out of the University of Colorado that was run across 6 cancer centers total. And one of my junior faculty, Dr. Elena Shaga-Sultanova, who works with me in the Young Women's Breast Cancer Program, took this on as her lead clinical trial, and we were able to get it started and launched initially through the fact that I had some lovely funding from donors who support my work, and we could move this forward. Elena was able to get a large grant. That's what the Aspire Award is that you see up in the corner. And that was a $1.4 million gift from Pfizer, a grant award to then launch the clinical trial based on the preclinical work Elena had done in my lab demonstrating the promise of this combination. So it's a 3-drug combination, all pills, one of which is tucatinib. The other 2 are standard breast cancer therapies for estrogen repositive disease. And the fun chart that you see down below that looks like a bunch of colored lines is the results of that clinical trial. And it's what we call a swimmer's plot. So the longer somebody has a line, the longer they've been alive and on their treatment. And the ones with the black arrows are still on treatment. And the importance of tucatinib is that it crosses the blood-brain barrier and can effectively treat brain metastasis in these cancers. And so this work's been presented and moving forward in the clinical trial pipeline. And I want to highlight, so young Hannah is a woman who found me through the Young Women's Breast Cancer Clinic. And when she came to me, she was 26 years old. She had not been aware that she had been feeling that she was quite as sick as she was. She hadn't been feeling well and had seen a few doctors, but they had really missed what was going on. And unfortunately, Hannah presented with very late stage advanced metastatic breast cancer involving many of her organs and was critically ill. So we were able to get her started on treatment straight away, and we got her out of danger. But unfortunately, about a year later, she developed a brain metastasis and some progression in the disease in her liver as well. So she joined us on Elena's clinical trial with this 3-drug combination, and she's doing fantastically well. She's that top line there where you can see the number of months she's been on treatment. It's actually out over 24 months now. We just haven't updated the slide. And she's gone back to work. She's looking forward to COVID ending so she can see her long distance boyfriend a little bit more often. Her brain disease is under fabulous control. So she's currently 29. And really living a very normal life. And the beauty of this is she takes all pills. She doesn't have to come into the infusion center and get any IV treatment at all. So, you know, this is a life-changing opportunity that we were able to give this young woman based purely on our own work at the university that began its journey supported through philanthropy. And so it's stories like this that keep us going, and it's funding from folks like you all that keep us going too. I'll leave it there. Next slide, Angela.
Okay, hello, my name's Haida Ford. So I'm going to introduce the more basic science, basic and translational science component of our talk today. So the three of us, and I'm going to let Jennifer and Tracy introduce themselves before we go on to the research, all are involved in basic and then translational science. So I'm a professor in the Department of Pharmacology, and I'm also the Associate Director of Basic Research for our Cancer Center. I hold the David and Margaret Shirley Groney Chair, so I'm very thankful to the Groneys for providing me with that endowed chair, which helps actually stimulate some of the research at the very early points before we get federal funding for it. And I think I'll let Jennifer and Tracy introduce themselves, and then we can each talk quickly about what we're doing. Jennifer, do you want to introduce yourself?
Sure. Hi, I'm Jennifer Ricker. I'm a professor in the Department of Pathology here at the Anschutz Medical Campus, and I'm involved in the Cancer Center as a co-leader of one of our research programs called the Tumor Host Interactions Program. And you'll hear a little bit about that as we talk about the basic science, but really it looks at how the tumor interacts with other parts of the body and that are not cancerous. And so that those interactions and crosstalk is what we focus on. And I'll tell you more details about my research in a bit. And I'll let Tracy introduce herself.
Hello, everyone. Thank you for being here tonight. My name is Tracy Lyons. I'm an associate professor in the Division of Medical Oncology, and I am actually part of the Young Women's Breast Cancer Program that Ginger described earlier, and I'll tell you a little bit more about the work that we've been doing in that program. I have been with the University of Colorado through my entire career since my undergraduate, greater than 25 years. And I really think it's just a wonderful place to be doing research. And I'm really excited about the things my colleagues are going to present to you tonight.
All right, Angela, you want to go to the next slide, please? Okay, so I'm going to tell you about some work that started in my lab that is now part of a larger program project that we're developing in large part through the help of philanthropy. So what we're really interested in and what the greater group, I've listed some of the collaborators here at University of Colorado are interested in, is tumor cell plasticity. And what I mean by that is we're trying to understand metastasis. And when you think about tumor cells that are metastatic, they need to navigate many different steps and be exposed to different environments in order to survive. And that exposure to the different environments, for example, these cells start in the primary tumor, they change characteristics so that they can break away from the primary tumor and get into the bloodstream. They then need to disseminate through the bloodstream, get out of the bloodstream, and then colonize at a secondary site, whether that is bone or brain or liver or lungs, and grow out there. And to navigate all those steps of the metastatic cascade, tumor cells need to take on very different characteristics. So what is required to survive at the primary tumor site is different than what's required to survive in the bloodstream, and is different yet again than what is required to survive at a secondary site. And so really what we're looking at, can you go forward, Angela, please?
Yes.
Maybe again. I'm not sure what happened.
There we
Okay,
go.
right. So my lab has been really interested in this for quite a few years, and this larger collaborative project that we're now trying to put together contains people who have expertise. So my lab has expertise in this plasticity, so the ability of cells to move back and forwards between different states that are required to navigate the different sites they'll end up in. And other labs in the group are, for example, have expertise in brain metastasis or in bone metastasis. And so what we're really interested in doing is looking at how the production of different RNAs, I'm sure you've all heard a lot about RNA from the COVID mRNA vaccines that have recently been developed, or the production of different proteins in these different settings. So whether you're at the primary site, in the bloodstream, or at the secondary site, and the rapid ability to go back and forth, whether we can actually inhibit that to inhibit metastasis. Can you move forward again, please, Angela? And so, yeah. And so what we're trying to do is not only understand on a molecular level, so at the RNA and the protein level, what those changes are that the tumor cells go through in order to survive all these different steps of the metastatic cascade, but then we're trying to figure out ways that we can actually target that. So can we develop novel inhibitors that will inhibit the ability of cells to be able to move between different states? And that if we can, we might be able to inhibit the various steps of the metastatic cascade. And if you go forward one more slide, Angela, we've recently, one more, sorry, yep. So we've actually just recently published on a novel inhibitor that we've developed through a variety of methods that actually shows that it dramatically can inhibit metastasis. So this inhibitor was given to these animals. This is an animal model only for 3 weeks. And then we followed the tumor metastasis out for a course of almost 8 weeks. And what you can see here is when we administer the inhibitor, if you look at that red bar, you dramatically inhibit the ability of these cells to grow at secondary sites to metastasize. And again, since metastasis is really the major killer of women with breast cancer, this is what we really want to target. And so our larger group, again, with the help of philanthropy, we've gotten philanthropic funds through the Cancer Center, that is actually helping us to now create a large team that can all work towards the end goal of inhibiting the ability of these cells to move between different states and therefore inhibiting metastatic breast cancer. And we're actually looking at this in the context of both hormone receptor positive breast cancers, as well as the triple negative breast cancers that you heard about earlier from Jen. And so again, without philanthropy, this larger group would never be able to come together to try to take this work, which is from the very basic science level, to the bedside. And so now I'm going to let Jen, I think, talk about her work.
All right, thanks, Haida. So I'm going to tell you a story that is a collaboration between myself and Dr. Anthony Elias, and my expertise is in hormone receptors. But we also, I wanted to mention quickly, similar to what Dr. Ford told you about just now, we also have another program project that we're developing, again, starting with help from philanthropy, that involves Dr. Borges that you heard from, and Dr. Lyons that you'll hear from next, and myself and an immunologist, Dr. Jill Slansky. So that project involves figuring out how tumor cells suppress the immune system so that they can avoid an immune attack, and how to reverse that or interfere with that. And Tracy will touch on that a little bit, Dr. Lyons, in a minute. But this story involves looking at hormone receptor positive breast cancers. And we started out with trying to figure out what happens when they are evading one of the most common therapies in breast cancer, and still one of the most effective, is to interfere with the action of the estrogen receptor. But despite how effective those are, some patients have recurring disease that does not respond to various different strategies to interfere with the action of the estrogen receptor. And so what we noticed in my lab, again, with the help of small donations and local groups like the Colorado Cancer League, and then we went on to get federal funding through the NIH, and then also a $5.9 million grant through the Department of Defense to look at this study, and that included 2 clinical trials. So in my lab, we did some studies similar to what Dr. Ford just taught you about in mouse models, And what we saw was that some of these tumors switched from being dependent on the estrogen receptor to now evading that therapy and becoming dependent on the androgen receptor, which you may have heard is a target in in prostate cancer. So Dr. Elias and I worked together and designed 2 clinical trials. And the one I'll show you more about today, because it just finished, is one in metastatic breast cancer. So ER positive metastatic breast cancer, in which that is persisting and despite all the traditional treatments targeting the estrogen receptor and chemotherapy as well. So we decided, based on our preclinical work and animal models, that they do become dependent on, some tumors become dependent on the androgen receptor. So we combined the traditional fulvestrant as it's an estrogen receptor degrader with an anti-androgen receptor called enzalutamide. So that interferes with the action of the androgen receptor. And I'll show you the results. And if you go to the next slide, Angela, the trial in patients with metastatic disease that was persisting despite the traditional therapies targeting the estrogen receptor or chemotherapy. When we come in with the combination of the fulvestrant with the anti-androgen, even some of the patients that have had prior fulvestrant are now responding. Their tumors we interfered with the progression of their disease by adding in this anti-androgen. So you can see here that some benefited even with highly metastatic disease that's been pretreated with many endocrine therapies as well as chemotherapy strategies, got up to a year in this swimmer plot of the progression-free survival. And so this indicates that we are seeing some efficacy. And now our trial that's currently still ongoing is in the neoadjuvant setting, which is in primary tumors that are locally advanced, large tumors. And we can see if we come in earlier with this combination, if we can get an even better response instead of this trial that you see the results of here, where we're dealing with highly metastatic and highly pretreated disease. So I am going to stop there and let Dr. Lyons tell you more about her work.
Thank you, Jennifer. So as I mentioned before, I've been with the university for a very long time. And about almost 13 years ago now, when I was finishing my graduate training, I partnered with Dr. Borges because she had made this observation about women being diagnosed with breast cancer within 5 to 10 years of recent pregnancy. And these patients were doing very poorly in her clinic. And I became really fascinated by this poor prognosis, especially given my training as a normal mammary gland biologist. I was really fascinated about the changes in the mammary gland that happen when a woman becomes pregnant. And so as Jen Diamond mentioned earlier, this image on the left is my inspiration. It is a patient who was 36 years old, diagnosed 1 month after the birth of her second child. And if you look at the green ring, as Dr. Ford very nicely introduced, when cells spread to different parts of the body, they must spread through the vasculature. So I think of these as roads out of town. And their first pit stop is often the lymph node. And I won't go into great detail about this, but the green ring that you see in this image is a lymphatic vessel. So it's like a blood vessel, but it leads to the lymph node as opposed to other vital organs. But it's thought that the lymph node is the first stop on the way to a place like the lung or the brain. And the brown cells that you see are expressing a molecule that our group has identified that makes tumor cells do pretty much all of the nasty things that Dr. Ford showed you in her lovely schematic. It makes them invade, it allows them to enter the vasculature. And as Dr. Ricker mentioned, it also suppresses the immune system. So we're really interested in how this molecule is working in cancer. And recently we've published a level of this molecule that can predict for metastases in our postpartum patients. But what we're really excited about, if you can go to the next slide, is we have in-house developed a novel monoclonal antibody-based inhibitor of this molecule. And we did this with 100% philanthropy. There's a family that has been consistently every year giving money to the Lyons and Borges labs. And this money was really special to me because I also know this person quite well. And so I was waiting for something really exciting to do with this money. And we used this to generate this monoclonal antibody, which we made 2 of them. So you can see MABH1, MABH7. So this is an animal study like Dr. Ford showed you, and you can see that the red line is the animals that received no treatment, and the blue lines are the animals that received the treatment. And one treatment worked phenomenally well, making the tumors completely melt away. And I can tell you, I don't have a swimmer's plot. I wish I did. But these animals then have gone out to 100 days without any reemergence of their disease, which is really a great measurement of how good this treatment is. And I will mention the arrows show you when the treatment was actually removed. And so these animals continue to survive with no evidence of disease. And so with that, I will move on to the next slide.
All right, so I guess I can jump in and just finish, and anyone else can jump in if they'd like also. So I think hopefully from what you've heard from all of us is number one, we have a team of breast cancer clinicians and researchers that work very closely together, all towards the same end goal, where we're hoping to make a difference, particularly in metastatic breast cancer. That philanthropy has really enabled our group to become more collaborative and more cohesive, and to work towards our common goal, which is obviously inhibiting, developing novel means to inhibit cancer progression. And throughout today, we've been talking about the fact that philanthropy has allowed our laboratories to work on new, often high-risk projects, high risk but high reward ideas. Tracy just described one beautifully that she couldn't have done without philanthropy. And that without this preliminary data, these larger grants that we've all gotten, Jen again described to you one that she got from the DoD, just wouldn't have been possible. A number of us also hold endowed chairs, and these endowed chairs allow us a more protected time to do research, to train the next generation of researchers, and also again to do something that's very high risk that we may not have funded by our federal grants. We've got an example here that Tracy just mentioned about the novel monoclonal antibody treatment for postpartum breast cancer, and that was generated really with 100% philanthropic support. And so we just want to thank you all and everybody, on this call for listening to hear about what we do today, and also thank all of our generous benefactors for what they've done for us here at the University of Colorado.